Plasmodiophora. Introduction. Plasmodiophora is the best-known genus under the family Plasmodiophораceae. The well-known species P. brassicae is an obligate endoparasite which causes club root disease of crucifers like cabbage, cauliflower, turnip, radish, etc. The disease was first reported in Scotland in 1780. After many years, detailed scientific work about the pathogen and disease on cabbage was done by Warrenan, 1878. Symptoms of disease caused by Plasmodiophora Brassicae. The symptoms are visible in the root of affected plants. The roots become abnormally increased and swell up at different regions due to hypertrophy and hyperplasia. In cabbage, the root bears malformation or clubs of different shapes, hence the disease is called club root disease. The development of lateral roots becomes suppressed. The drooping of the leaves during sunny days, with gradual progress of disease, leads to permanent wilting. The disease-infected tissue of root cabbage shows some variations in symptoms in different hypertrophied cells containing plasmodia or spore hosts. Masses. A. Vegetative structure. Vegetative mycelium is of three types. Primary mycelium. It originates by the germination of uninucleate basidiospores carrying either plus or strain. The cells are uninucleate, i.e., monokaryotic. It is short-lived and becomes binucleate by fusing of two compatible hyphae. Secondary mycelium. It originates from primary mycelium. After fusion of the hyphae of two opposite strains, the nucleus from one hypha migrates to the other and later gives rise to the binucleate secondary mycelium, i.e., dikaryotic. It is long-lived and abundant. Tertiary mycelium. The secondary mycelium A. Monokaryotic hypha grows extensively under the soil and becomes organized B. Dikaryotic hyphae special tissue to form the fruiting body or basidiocarp. B. Fruiting body or basidocarp. The fruiting body appears like umbrella above ground. It is made up of dikaryotic hyphae. These hyphae are called tertiary mycelium. The mycelium is subterranean. The hyphae are septate and branched. The cells communicate with one another by means of a central pore in the septum. It is a typical dollopore septum. Sexual reproduction. During sexual reproduction, each nucleus of Gametotholus accumulates some cytoplasm around itself and behaves as an individual unit, called gametangium. The nucleus of each gametangium undergoes mitotic divisions and forms 4 to 8 daughter nuclei. Each daughter nucleus accumulates some cytoplasm and behaves as an individual unit. Each unit develops into a spindle-shaped, biflagellate gamete. Gametes are alike, smaller in size, and are called isogametes. The gametes are liberated out by bursting the gametangial wall and root hair cells. The gametic union, only plasmogamy, takes place in the soil, results the formation of binucleate and quadriflagellate structure. Secondary phase. The binucleate and quadriflagellate structure then enters again into the host tissue, initially through epibulma towards the meristematic zone. It retracts the flagella in contact with the host tissue and after entering inside host cell it becomes a moeboid, called myxamoeba or plasmodium. The myxamoeba can spread from cell to cell through the cell wall. Initially, it remains in cortical cell and later invades to the other tissue. Within the host cell, the myxamoeba enlarges and forms a naked multinucleate plasmodium by repeated division of its haploid nucleus. At maturity, it almost occupies the entire lumen of the host cell and surrounds the host nucleus. Later on, the infected cell undergoes hypertrophy, i.e., enlargement of the cell, following hyperplasia, i.e., repeated division of cells. Thus several cells are formed and the internal plasmodium also distributed to each cell by fragmentation. Consequently, the neighboring uninfected cells get stimulated and undergo hypertrophy and hyperplasia, thereby tuberous swelling or malformation takes place. Due to rapid growth of club, cork layer formation in root becomes hampered, thereby it gets infected by soft root bacteria or fungi. Due to secondary infection, the decay of the club leads to the formation of toxic material, which causes wilting of the plant. Spore formation. After maturation of the haploid plasmodium inside the host cell, the haploid nuclei get associated in pairs and karyogamy takes place at this stage. Thus many zygotic two-end nuclei are formed. The diploid nuclei then undergo meiosis and form haploid nuclei. The cytoplasm undergoes cleavage and accumulates around each nucleus. The entire protoplast is then transformed into uninucleate small bodies, surrounded by smooth wall, called resting spores. The mature spores are set free, after decay and decomposition of club root. The spores can survive for a long time in soil. During favorable condition, the spores germinate and produce zoospores.